And it's widely used by molecular biologists to predict primer specificity. And it's this last point that we want to talk about a little bit. All right, so what's wrong with using BLAST for predicting cross-hybridization? So when it comes right down to it, the main disadvantage of BLAST is that it searches based upon sequence similarity, not sequence complementarity, which is what we're interested in if we're interested in scanning a primer or probe against the genome database. Now, because BLAST cannot search for sequence complementarity, people will commonly use a workaround, and the workaround that they will do is they'll say, well, gee, BLAST can't scan for the complement, so let's just take the complement of the oligo that I'm interested in and then use BLAST to see if BLAST can find similarities to the complement of my oligo. Now, that procedure right there leads to 5 problems, at least 5, and we're going to elucidate each one of those and why they're problematic. So the first one is that BLAST gives the wrong ranking of hits. And that's, again, because it's scoring based on similarity rather than thermodynamics. I'll have a whole slide of that in the next slide. Because of the incorrect ranking of the hits in the wrong scoring method, BLAST misses approximately 80% of the thermodynamically stable hits. And we'll show some examples of that. Another problem with BLAST for determining cross-hybridization is that if you blast oligonucleotide against a large database like NT, you actually get too many irrelevant hits. So for example, if you were trying to design a primer against the human genome, humans are mammals, and so that primer would be likely to hit other mammals like the horse genome or the pig genome or the cow genome. So any one of those would be evolutionarily related but have no relevance at all to a primer design. And so you get too many of the hits you don't care about. So the point 2 indicates that BLAST misses a lot of the hits that do matter, and it also misses a lot, or also gives you too much of ones you don't want. Furthermore, BLAST does not distinguish between a hit that is extensible versus one that's non-extensible by a polymerase. And we'll see that that's very important for the design of primers versus probes. All right, and lastly, BLAST has no mechanism to detect amplicons. So even though you might get a primer that binds somewhere in a genome that you care about, what matters is, does it form a false amplicon? And ThermoBLAST actually incorporates an algorithm to detect such amplicons. And that becomes extremely important when you're doing multiplexing. So if you have many primers, then the chances of 2 or more of those primers coming together to form an amplicon becomes much higher. All right, so let's talk about the ranking of results. So I mentioned that sequence similarity is not the same as thermodynamic stability. And we can see that right off the bat. You know, BLAST scores all sequence matches the same. So an A match is the same as a C or G or T. But we know in thermodynamics that GC base pairs are not the same as an AT base pair. GCs are much more stable. And furthermore, really you need a nearest neighbor model to accurately predict hybridization. All right, so off the bat is a problem with using the complement and then using BLAST. Another problem is that BLAST scores all mismatches as if they're the same. So BLAST does not understand that some mismatches are stabilizing, such as GG, GT, and GA mismatches are all thermodynamically stable mismatches versus mismatches that are destabilizing, like AC, TC, and CC. And these range over a factor of over 1,000 in equilibrium constant. So it's a big effect that not all mismatches are the same. Furthermore, BLAST uses an incorrect model for bulges. Well, it's correct model for the gaps that are found in a split alignment, but it's incorrect if you're doing it for complementarity with bulges. All right. In addition, BLAST does not include the effects of dangling ends or different sequence types. So hybridization involving DNA versus RNA are not accounted for, salt and temperature effects, and multiple mismatches. All of those are considerations that make the scoring of BLAST compromised for the application of finding cross-hybridization. Now, furthermore, maybe the most important is that BLAST has a minimum word length. That word length means that every hit must have at least 7 consecutive perfect matches. If there's even one mismatch in that window of 7, word length of 7, then BLAST will completely ignore the hit. And that becomes extremely important in finding all of the thermodynamically stable hits. All right, so here's some of an illustration of some of the motifs that are neglected by BLAST. So the dangling end. So here is here is a short oligonucleotide hybridized to a much longer genomic DNA. And it's not just the pairing regions that matter. It's these extra dangling ends also make a contribution. In addition, mismatches are not only different than each other, but they depend where they are in the sequence. So, excuse me, a mismatch in the middle of the sequence is not the same as a mismatch at the very end of a sequence or near the end, at the penultimate position. All of those effects are taken into account by Thermo Blast, as we'll see later. And here's some examples of hybridization events that would be missed by BLAST because of the short word length. So we have a very long complementary section of DNA, and the red regions are mismatches. Well, if you look at this sequence, you notice it's extremely thermodynamically stable. The fact that there's no stretch that's longer than 7 nucleotides means that these would all be missed. All of these are relatively stable, and that's due to that word length. All right, so now let's talk a little bit about the ThermoBLAST Cloud Edition. We just released this for full public release. We did a 2-month beta testing period with some of our longtime customers who gave us a lot of good feedback, and we've responded to that. But ThermoBLAST Cloud Edition combines the speed and database capabilities of BLAST with a lot of extra goodies. So one is that it includes thermodynamics-based scoring instead of similarity-based scoring. We've completely rewritten the seeding and extension algorithms that are in BLAST. They're really completely different, how we do the algorithm for finding the hits. We've added something called genome playlists. I'll give you a whole slide about what that's about. Massive cloud computing to allow for quick computation times, and a new genome viewer application that allows you to basically do a reality check to see if the primers you've designed are binding to the intended site, where you can see the GenBank annotations in a genome viewer. And then lastly, automatic detection of the Amplicons. So in summary, the ThermoBLAST ranks hits based on affinity rather than similarity. So it's a totally different model. All right, so let's talk a little bit about genome playlists and what that's about. So the best analogy we could make is to compare ThermoBLAST to iTunes, which I think a lot of people are familiar with. So in iTunes, the smallest unit of songs that you can get is a single song. For us, in ThermoBLAST, the equivalent would be a single GenBank accession, which contains a single DNA or RNA sequence. A collection of songs into an album, a small collection, in iTunes would be called an album. In ThermoBLAST, a small collection of songs would be like a genome, for example, combining all the sequences found in the chromosomes. For example, a human genome has 22 autosomes and an X and Y chromosome. So the combination of all of those is a small playlist that we would call a genome. And lastly is the concept of a playlist in iTunes, which is a collection of songs and a collection of albums. So we similarly have a collection of such accessions or other playlists that we can make in ThermoBLAST. And we'll see why that's valuable. So one of the nice features about ThermoBLAST is we've already preloaded our website portal with a whole bunch of playlists that many of our users have already told us have been very useful to them. So we already have the human genome, latest version of the human genome, human microbiome. All 43,000 GenBank accessions are preloaded into a playlist. The entire human RefSeq, which is about 98,000 GenBank accessions. We've assembled a bacterial diversity panel. This is a panel of about 1,200 complete bacterial genomes which are useful for finding false positives, and then also playlists for soil microbes. I'll show a more complete list than this later on. Now, one of the nice features about these preloaded playlists is that you can mix and match them and combine them into larger playlists, and you can add your own customized playlist to those. So even sequences that are not found in GenBank, if you have your own proprietary sequences, for example, you can make your own custom playlist and combine them with the playlists that are available in GenBank. So that gives the user a lot of flexibility and allows them to do things such as making inclusivity panels. So, for example, if you want to design a set of primers that bind to all of the sequence variants of Ebola, and yet not bind to anything else, then you can make an inclusivity panel including all the known Ebola sequences and make an exclusivity panel against everything that you would not want those primers to bind to. For example, you would want Ebola primers to bind to any influenza A variants, which are a common thing seen in the clinic. All right, so this is the concept of an inclusivity versus inclusivity panel. I'll be showing you a demo that shows some of this. So, for example, if we made an Ebola playlist, an inclusivity Ebola playlist with all 144 known complete Ebola genomes, that's all the ones that are in GenBank. That's about 2.7 million nucleotides worth of genomic sequence. We can determine if a set of primers, how many of these 144 genomes are hit by a particular set of primers. So we can determine the coverage of, how many primers, how many of the genomes our primers actually will amplify. On the other hand, we want to avoid false positives with our primer designs. And so we might be able to come up with a quite long list of non-Ebola background sequences that could give a false positive to an Ebola, such as the dengue fever virus. All right, those are genetically related to Ebola, and so you might guess that a poor design of an Ebola sequence might hit some of the dengue viruses. Many patients who present in the clinic for Ebola, what looks like Ebola symptoms, influenza A symptoms look quite similar. So we would want to make sure that whatever primers we design don't hit influenza A or B or other fever-causing viruses like Marburg or Lassa fever or any of the other ones. We want the human genome would obviously be present in a sample taken from a human to test for Ebola. So we want to make sure the human genome doesn't cause a false positive. Any of the microbes in the human, like the human microbiome, or contaminating soil microbes. So all of those can be used to determine if you have any false positive possibilities. All right, so with that introduction, I'm going to be moving into a demo in just a moment. I just wanted to briefly show you some screenshots from the software. The results are going to be shown after the user chooses their playlist and enters in their primers. They'll get a results page that looks like this. It will have a list of individual hits for each primer, and it will have a list of amplicons that it detects. So I'll show you more examples about that. I just wanted to mention that also in the output it will show us a genome viewer where we'll see the forward and reverse primers and any probe, and you'll be able to click on and see what the genes are that those are hitting. There's also a sequence view down here. You can expand it if you want to see more genomic context. There's also a structure view that allows you to see if your primers are indeed hybridizing the way you intended. And there's a feature to download Amplicons, including extra sequence if you want. All right, so this brings us to the demonstration. If you would like to sort of poke around and look for yourself, you're welcome to do that. Here's the website for our company. And just click on login here, and it will ask you to register. If you register, you can sort of see what we got. All right, so I'm going to go back, and I'm going to escape out of this and go to our company homepage here. All right, let me full screen this just so you can see it. All right, so this is our company homepage, and this is the login for the portal. Let me go to that. I'll go ahead. I've already registered. If you haven't registered, you can just click over here to register. So we'll go ahead and log in. and it brings us to this Let's Get Started page. So this is the entry into our portal. Currently we have two products on our portal. One is the copy count product. This is a product that allows users to take their already existing qPCR data and analyze it to get absolute copy number. That's a product we won't be talking about any further today. We've already posted a webinar about that if you're interested. There's a product called Thermo Blast Cloud Edition. That's what we're talking about today, and I'll click on that in a moment. But I do want to take this opportunity to share with you that we're continuing to build out our portfolio of products. And in the future, we'll be adding to this portfolio product for primer design and multiplexing, genome enrichment, things along the lines of epigenetic detection, antisense therapeutics, siRNAs, etc. Okay, so for now we have this Thermo Blast Cloud Edition, and we'll show you about that. So just click on this button, click to check for mishybridization, and it brings us into the welcome page for Thermo Blast. And there's two things. You can either view some old results that we've previously run, or we can run a new job in Thermo Blast. And there's three basic steps to running a Thermo Blast job. One is we're going to pick the collection of targets that we want to Thermo Blast against. All right, these are our playlists. Next, we'll want to choose or enter in our primer and probe sequences. And lastly, choose our salt conditions. So let's go ahead and get right into it and show a run here. All right, so I clicked on that, and it opened up a page. The first step is that we have to choose our playlists. So we have a list of most popular playlists. There's 3 tabs here. If I scroll down here a little bit, we'll get to see a little bit about ones that are preloaded. So we have one here for the HIV genome. Here's the most recent version of the human genome. Here is a list of the Ebola virus inclusions of 144 sequences. Here are some medically important bacteria. All right, so list of, I don't know, 70 or so bacteria. This playlist here of all viruses has about 65,000 complete virus genomes that have been sequenced to date. Here is the human RefSeq with all 98,000. So you can see there's a whole variety of these panels that we've already preset in here. If anyone has any questions about ones we've already set up, feel free to ask about those. Over here is a feature where if you click on playlists you created, it will show you some of the custom playlists that either you own or you shared with other users. All right, so over here I made my own private playlist for the mouse RefSeq over here. And I did one for zebrafish, and here's one for Ebola viruses and other viruses, all right, dengue fever viruses. So I made a variety of, and there's more in there, influenza A viruses, etc. All right, so one of the things you might be interested in knowing is, what do I do if the playlist I want isn't there, or maybe I have a proprietary sequence? How do I go about doing that? Well, it's very simple. You just click on that button to create a new playlist. And then here you give your playlist a name, like John's Playlist. All right, so we'll make a new one. All right, I've never made one named that before. And then over here, if you hit Add Content to this playlist, we have a variety of ways in which you can add sequences to your playlist. So one is you can search NCBI to find a playlist of interest. Another is if you already have a list of accessions, you can click on Enter Accessions, or you can upload a FASTA file. Or you can combine it with playlists that you previously done. Let's go ahead and do it with entering by accessions. All right, so we come up with this window here, and let me go to an Excel spreadsheet here. Here's an Excel spreadsheet just for purposes of illustration. This has whole columns full of GenBank accessions. Let's say I wanted to make a playlist for HPV cancer types. All right, cancer-causing variations of the human papillomavirus. So I can select those GenBank accessions and just come over here and paste them in. And you can paste in a lot. We've pasted in up to 100,000 accessions and got it to take it. And you just hit Check Accessions. What that's doing right now in real time is it's going to NCBI and checking if those are valid accessions. If you made a typo or something, it would tell you there was something wrong. And there's a couple of buttons that you have to click through to process those. All right, so maybe I should have given this a better name, like HPV cancer. That might be a better name for this playlist. All right, so it shows you the playlist that it found. So there's human papillomaviruses type 16, type 18, type 33. Those are the really important ones that are cancer causing. You can see the GenBank accession numbers here for each of those. You can download the actual NCBI if you want by clicking. If there's any of them that you particularly don't want, you can hit remove to do that. All right, so down here we need to save the playlist one more time. And lastly here. Okay, so at this point, the server actually has to ping NCBI and go through the process of building that playlist, which takes a little time. It's not because of our software, because NCBI has a limit. They can only download 3 sequences per second. All right, and they place that limit on all users. But essentially, we just wanted to show you the flexibility of being able to load in a new playlist. All right, if I come down here, I should be able to see that new playlist. Here it is, HPV cancer playlist, and it's currently preparing it. So that's why once it's ready to go, you'll see a notation like add this playlist to your current run. It'll take a few minutes for it to do that. All right, so in the meantime, let's go ahead and give you a sense of how the program actually works. I want to get you to at least run one all the way through. Let's go to the most popular playlist for a moment. Let's go ahead and run against this Ebola background. All right, so I can add this playlist to my current ThermoBLAST run. And that contains 144 complete genomes of Ebola. It takes a minute here to communicate with the portal here, but it's going to come up in this window here. We're going to get a little basically summary here. There it is. It popped in. So Ebola background sequences. All right, so with that, we'd be ready to run it. If you're interested in knowing what's in there, you can click on the View playlist down here. If you do that, it takes us out of that window briefly and shows us what's in there. 144 sequences in that playlist, and here they are. Okay, so it's got a whole bunch of variants of Sudan and Zaire and lots and lots of. You can view all of them if you want here. So you can see there's, you know, Côte d'Ivoire and Reston and all the different variants of Ebola. It's 2.7 million nucleotides. All right, so if I go back to playlist here, I can see my current job. Here it is. Let's go ahead and show you the work just so you can see how the full process happens. So I've chosen my background genome the playlist that I want to scan against, and I could have chosen a whole bunch of them here if I wanted to. But let's just move ahead here. So let's run ThermoBLAST against this background. All right. Then here we can either enter in our sequences manually, or we can enter in the sequences in batch mode. All right. So I'll show you in batch mode first, and then I do want to go through the exercise of showing it manually as well. So if we go to batch mode here, I already had this already made here, so I need to find it though. Here we go. So I have a set of primers here called the Ebola primers. All right, so these are some primers I just designed quickly. So there's a forward primer, there's a probe, and a reverse primer. This is a TACMAN design that I did quickly. All right, so the first part of this is just to enter in your sequences. If you wanted to enter in another sequence, let's say I had some other sequence, like I'll just say made up. So here's a new sequence. I'm going to type in some random sequence here. All right, so there we go. There's a random sequence. And then down here, I can either choose to make that a primer or a probe. I'm going to, for a moment, show you what would happen if you check. If you check primer, it automatically declares it as a DNA, since all primers are made of DNA. But if you would have chose probe here, and then the strand type, there'll be different types of strands that it will allow you to choose. In this case, DNA, RNA, or peptide nucleic acids. And that depends on what the makeup of is of the genome that you're going against. All right, so let's go ahead and show this as a primer. I could add that in just to show you. There it is. Here's my made-up primer that I just randomly typed in. All right. So with those primers, so I mentioned there was 3 steps. The first step was choose our playlist. The second step was enter in your primers, which you can do manually or in batch mode. And the third and last step is to choose your hybridization condition. So we can provide a target concentration, and we can provide a kit type. So we have a variety of pre-made kits here. Typical endpoint PCR or TACMan real-time PCR. These fill in these variables over here for the annealing temperature, sodium, magnesium, etc. You can also, let's say you have some exotic conditions that you're doing your hybridization under. You can define a new kit over here. All right, you can give it a name, etc. Like I could give it, John's kit. Oops, maybe I spelled it wrong. Here we go, John's kit. And we can give it a temperature, like let's say it's 58 degrees and some exotic sodium like 0.153, some weird number. And then magnesium, maybe that's unusual. That would be too high. All right. And then we'll fill out, it doesn't expect you to have a value for all these other ones. It can support all these different denaturants and buffer additives. If you have them and know them, you can put them in. But that's it. We can just go ahead and do that. Now I have this new John's kit, and that's permanently saved as one of the choices here in our list. Okay, so choosing buffer conditions, that's that. And then lastly, you just hit run. Okay, so that's it. The job is running. So really as simple as choose your playlist, enter in your primers, choose your solution condition. If you want to check on the status of your job, it comes to this. It actually automatically came to this if you don't click on that button quick enough. But here is the job that's currently running, Ebola background, other organisms. Oh, no, that's a different job. All right, at any rate, no, this is the one I just ran. I must have selected one different than the one. I was thinking that was the Ebola inclusion. I must have accidentally chose the background. At any rate, it's running. Let me show you while that's running some results from some other runs that I've ran, just to show you the sort of thing that this does. If I click over here and look at some results, it will show me. All right, so down here it has a list for each primer. It gives the individual hits for that primer. So here's forward primer 1, and here's each of the targets that it hit and its melting temperature for those different targets. All right, and there's a long list of those down here. Right, you can scroll through them all day if you want. There can be thousands of hits there sometimes. Up here are the Amplicons that it found. So it took each of the individual hits, and then it found all of their positions in the genome, and it looked for all cases where there was a forward hybridization event and a reverse hybridization event that were within some window. In this case, the window is 3,000 nucleotides. If those two oppositely pointing primers are within 3,000 nucleotides, we call that an Amplicon, and we would display it. Now, in this case, we were running this against the 144 different Ebola sequences. We have a nice feature here that allows you to download all of those Amplicons here, so you can see the all the hits in an Excel spreadsheet, which we can do just by clicking this. All right, let's see if that did it do that. I don't know. I don't see it here. My browser is. Where did that go? Anyway, that downloaded Amplicons is in my download file, so I'd have to find out where my download directory is. I think I have one of them open over here, just to show you. All right, where's our webinar? I wanted to show you what that looks like if you were to download. It looks like I didn't save it here. All right, that's all right. Let's move forward and just show you some of these other views for a moment. We have here a Genome View. Let's take a look at that. All right, so this is what it shows you for a genome view. You can see the locations of these primers. Here's the two blue ones are primers. Reds are probes. All right, so if you hover over those primers and probes, it tells you what they are. So this is the forward primer here. Gives you some information about the hybridization of that forward primer. If I hover over here, it shows me information about the probe and information about the other primer. And the intensity of these is actually related to how strongly they're binding. This reverse primer here is a much weaker hybridization. If I expand here, I can actually see what direction the primers are pointing in. So one's a forward primer, the other one's reverse, and we use this little red icon here to indicate a probe sequence. And down here are the GenBank accessions. All right, so if you hover over those, this first one is a CDS, a coding sequence, and in particular, these primers are binding to an L gene, one of the late onset genes from Ebola, and then that gene product is encoding of the polymerase gene, all right, the polymerase protein. So these primers are binding in the polymerase gene, which is highly conserved among Ebola. Down here is the sequence view, so you can see where those primers are binding, where the probe is binding. And you can select sequence from that region if you want to copy and paste. All right, if you zoom out, you can sort of see more of the genomic context here that that's within. Let's see if this works. Here we go. So here's my primers up here. Here's my late gene. All right. In that neighborhood is the viral protein 24, glycoprotein, et cetera. All right, so you get some idea about the genomic context of those hits. If I click on structure view, it shows me the actual structures of the hybridization events. All right, so here's one of them. Let's see, I gotta figure out how to do this, Joe. All right, so you can see the 3' end here. I don't remember how to blow it up or to. There we go. So you can make it so you can see more of the hybridization or less. All right, so you can actually see, is my 3' end actually hybridized? If you don't like the way, if it looks a little wavy, you can just grab a nucleotide here. It will optimize the picture to make it look straighter for you. So one of these is the forward primer. One of them is the reverse primer. This one's the reverse primer. Over here is the forward primer. All right, so again, we can see the 5' and 3' end just panning on those pictures. So you can get details about your particular structure, gives the melting temperature, delta G, up there. And then lastly, over here, we have our download Amplicon feature. If you click on that, it will allow you to, if you click on this, it actually is already done a Control-C, a copy command to copy the send strand. If you wanted to get more than just the actual Amplicon itself, you can plug in here a padding value, like extra 100 nucleotides, for example. That will put an extra 100 nucleotides on either side of the Amplicon. And this can be very useful. You'll see as soon as I click down here, we'll see it go from 208 to 408. Yep, it went to 408 because it's the 208 nucleotides of the Amplicon plus 100 nucleotides on either side. And that's very useful if you're trying to do primer design of a particular region, for example. Use some other software. All right, there are some other nice features I would like to show you. I need to be able to get to my downloads. How do I get to that? Let's see if I can get to it here. Here we go, downloads. All right, so when I clicked on that button here, oops, I'll close this. How do I close that? All right. Download Amplicons as a CSV file. That's a very nice feature. If I go here and click on that, I can see those, every single one of those hits. Now in this case, there was 144 genomes that were present in there. Right? I can open this up just to show you. These are different variants of Ebola here. And we can count how many are present. In this case, there's exactly 125 Ebola genomes that were hit out of 144. That's a pretty good set of primers that it would hit that high of a percentage. 125 out of 144. In fact, probably those last remaining 19 genomes probably have sequencing errors in them because this set contains everything, including sequences from 1976, the original outbreak. But so this is in the inclusivity set. You can see how well your primers that you designed, do they hit everything that you wanted. On the other hand, if you had an exclusivity panel, you want to make sure that the primers that you designed don't hit things that you would cause the false positive. And all of those would have been present in this part of the Amplicon hits up here. If your primer set hits anything in your background, you would see it there. All right, I think with that, I've pretty much gone through all the features about ThermoBLAST. And at this time, I'm going to turn it over to Joe to talk about pricing, and then we'll take questions from the audience. All right, Joe.
All right, thanks, John. So we offer ThermoBLAST Cloud Edition in 3 different pricing tiers. The first of which is the basic seat license. This includes 1 user login credential, full access to all the features John just discussed in ThermoBLAST CE, a limited number of submissions per month, 30 submissions per month, with a playlist size of 4 billion nucleotides. To put that into reference, the human genome is 3 billion, so that would allow you entire bacteria diversity panels and other playlists along with the human genome. And all these submissions will be run on the Amazon Web Service Cloud. Seat license starts at $999 a month, or it can be purchased for $9,995 annually. The next offering is the Pro Seat license model. Again, this is for 1 user login credential, full access to all these features in ThermoBLAST with 100 submissions per month, a playlist size of 20 billion nucleotides. Again, these submissions will be run on the AWS cloud, but there's larger volume, there's larger playlists and volume discounts available for multiple users in this Pro Seat license pricing tier. During our beta offering, we heard some feedback from customers in terms of privacy and concerns regarding privacy. So we offer an enterprise license as well. We've offered private Amazon web instances for some of our customers to mitigate this concern. We also can offer a local installation. This would allow complete user control of your own data. This is the highest level of security we can provide. Again, you'll control your number of CPU instances, the number of submissions, and the playlist size. So this puts the control in your hands. These licenses are quoted per customer. Okay, so at this time, I'd like to address questions from the audience. I'll just please continue to ask. I've received some as we've gone through our presentation, but please continue to ask. There's some good questions. The first of which is a good question. How does ThermoBLAST Cloud Edition compare to the version of Visual Amp in terms of speed and size?
All right, well, the version of ThermoBLAST and Visual Amp requires that you load any genomes onto your desktop computer. That step of getting the actual genomic sequence on your desktop and formatting it has been problematic for many of our customers. It does work, but it's hard to assemble certainly large playlists of genomes. Also, you're limited by the single CPU on your desktop, whereas the Cloud Edition has 16 CPUs, and we have multiple instances of 16 CPUs. And we're actually planning to increase that to 32 compute cores running on each ThermoBLAST job. So the speed is just way, way faster. You can just imagine much, much more difficult things. For example, we had a customer we did a biodefense panel for that wanted to design a 30-plex, so it has 60 primers, and they wanted to run it against everything in the kitchen sink for background. All right, so they had essentially every bacterial genome we could get our hands on. We had over 2,000 bacterial genomes, the human microbiome, a series of medically important bacteria, eukaryotic pathogens, etc. So it was a huge, huge playlist. And they wanted to make sure that essentially the primers they were designing for a biodefense panel against 30 different bacteria didn't give any false positives for other bacteria or other pathogens. So the cloud edition is much, much more flexible. Of course, it has the genome viewer now and several other features we've actually improved a lot of the algorithms as well, although we're updating that in our visual as we speak. I think that answers that question.
The next question is, the default playlist appears to be DNA. How does one enter an RNA playlist?
Oh, that's very simple. When we're choosing the sequences, it automatically, if you give them as GenBank accession numbers, then it automatically assigns the sequence type based on whether whatever GenBank is set it to. But you can change it. That's in the software there. It's probably not so easy for me to get to it from here. But it's very simple to set that. Let's see if I can find it quickly for you. All right, so if I did new project. And then I went to playlist you created. All right, just reviewing again the mechanisms of creating playlists. Here we go. You have to click on create a new playlist here. And from within that, once you've created a playlist, it'll be in here, and you can change. It'll show you the strand type, but you can change it if you think it's a single-stranded or double-stranded, if it's DNA or RNA. For playlists, we only support DNA and RNA since that's all that's present in biology. But for the primers and probes, we support a variety of strand types. In fact, we have more than what's shown here. We're going to be updating the portal very soon to include morpholino, PNA, 2'-O-methyl, phosphorothioates, and a variety of point nucleotides like LNA, and those things will be coming later on. So we do support a variety of those as well. But that's where you do it, right there. It's just enter in the strand type.
Okay, the next question is, will the search report multiple hits? For example, Amplicons in the same sequence.
Yes, it does. They would each be reported separately. And that's particularly important when you're dealing with a large genome like the human genome. So, you know, you may desire your particular set of primers to go to the X chromosome, but in fact, it might bind to some other chromosomes in the human genome. And or even within the X chromosome, it will show you all of the hits that are found. As long as they're extensible, they will be shown as individual hits. If they're not extensible and probes, it will show those hits. If they form amplicons, it will show all the amplicons. So the answer to that question is basically yes.
Okay, the next question is, can I run ThermoBLAST CE against the NR database?
No. And we do not recommend running against NR in general. You know, you have to remember what the goal of ThermoBLAST is versus what it is when you're doing BLAST. When you're doing a BLAST search, you have a new sequence of unknown function, and you want to just blast against everything out there to see what the heck it is. With ThermoBLAST, it's very different. We have a case where you have a set of primers, and you want them to amplify or a probe that you want to bind to a specific genome, and you're worried about the kinds of things that might give a false positive. And that's not everything in GenBank. NR would be very inappropriate. You get lots of hits you don't care about. As I mentioned, if you were trying to design a primer for a human gene, well, NR would give you things against the horse and chicken and cow and all kinds of stuff you don't care about. Whereas with our playlists, we're allowed to make focused playlists that have the genomes that actually could cause a false positive in your assay. And this prevents, you know, information overload, which is what you get with BLAST. So you get a lot of hits you don't care about.
Okay, the next question is, can I import my own sequences? And if so, what format type?
Yes, you can import your own sequences. We currently support FASTA format. So you can actually just make a text file. I think I have an example over here. Let's say you have some set of sequences. Let me find this for you. Let's say you have a set of sequences that, I'm going to have to search my desktop here for a moment, that have not been deposited into GenBank. Maybe you're proprietary secret sequences, you know, you're in the military or something, highly, highly top secret. All right, so you make up your own set of sequences, let's say. I have a set here. So all you have to do is just make up a FASTA formatted file. So you give it the usual greater than symbol with a annotation, and then your sequence, and then your next annotation and sequence. And you can put as many sequences in there as you want. If there's an NCBI accession found anywhere in the annotation line, then our software actually recognizes that, and it will ask the user, would you like me to go to GenBank and get the latest version of the sequence, or do you want to use the version that's actually in this text file? And it will do either one. If you don't give it an NCBI accession, then it will just assume that the sequence that follows is the sequence that you want. So you can very, very easily input your own. That was one of the choices in that. Let's see if I can go back to that. Under the add content to this playlist, one of them is upload FASTA file. So if you select that, then it will allow you to browse your desktop and find a FASTA file. Let's see if I can find that one. There it is. So I just choose Ebola 19 sequences, uploaded them to the cloud just that fast. All right. Now, in this case, I had accessions in that playlist, and so it says, you know, do you want me to actually go to GenBank and get fresh sequences, or do you want to use the ones that are actually in there? If you hit yes, it's going to reload them in. If you hit no, just use the sequences as they are in the file. All right, so if you hit no, it would actually use it. So that's basically the answer to that question.
Okay, do you have any publications for ThermoBLAST CE in peer-reviewed journals?
We haven't published on ThermoBLAST Cloud Edition yet because it just came out a month ago in its commercial release. But we have had a lot of publications on ThermoBLAST, cite the use of ThermoBLAST, specifically when it was incorporated with Visual Amp and Developer Edition. So one of the notable publications is one that we did on a design of primers and probes for the 2009 pandemic H1N1 influenza that was published in the Journal of Clinical Chemistry. And then most recently, just this last year, Norm Watkins from our company collaborated with a group at the CDC to find a set of primers for tick-borne, was it West Nile virus?
Yeah, it was Lyme disease and
Lyme disease, sorry. Yeah.
But other tick-borne viruses, the Journal of Medical Entomology. But if anyone has specific questions for publications, we have some on our website, but I can follow up, and I have those in a PDF form I can send.
We've also published a little paper about the 7 myths of PCR design. It's getting a little old now. It's from 2007, but it has a lot of information about BLAST versus ThermoBLAST, about the sort of guts of how we do what we do. All right, let me take the next question, I think.
So the next question is, how would you batch upload in Excel for high throughput screening? So batch upload primers into ThermoBLAST CE for high throughput screening.
Oh, I already showed that actually. Maybe I went through it very quickly. Let's see here. If I go to new project, yeah, I actually did show that. So if you just chose a database quickly here, we have to go through the 3 steps. Let's just choose like this, HIV virus, just to get to the next step. I think I clicked. All right, here's our current job. It takes a minute for it to upload that. But basically, we already showed how to batch mode. Once you've entered in your sequences one time, you can save that CSV file that has the primers in it, and you can upload it at any time. Yeah, here we go. So if I hit run against that playlist, and over here, you just, right here, you can batch import your primers and probes here. And if you're interested in the format of those files, we have some example CSV or Excel files that you can download that show you, what columns have to have the primer name, and that sort of thing. All right, any other questions?
It looks like it.
All right, everyone. Well, thank you for joining this webinar. If you have some burning questions that you want to email to us, I would be happy to answer them. I'm sure Joe would be happy to answer them as well. And once again, we thank you for your time and being able to show you about